Hello, today my name is Gregor Mendel. I am the father of genetics, and I am going to be telling you about patterns and process of inheritance, also known is possibly called Mendelian Genetics. How interesting to have a whole field of biology named after you. I am also going to apologize about my accent. I'm sick today, and my Austrian accent is a little bit off. So forgive me if it sounds like 5 or 6 other languages. I'm doing my best to talk to you today as it's very cold and snowy, and I'm also pretty sick. But why is inheritance important? If you look at the birds on the right, what you will see is that one of the organisms has very striking coloration, and the other organism of the same species is a little more conspicuous than the one on the right. And the one on the right is a male cardinal, and you could see the striking reddish and blackish coloration, which is designed to show and display a certain level of fitness for that male. Because if that male is still alive after having colors like that in such a white background, then it must be fit enough in order to carry the genes that the female wants to transfer to her little birdies. If you look at the female is designed to be a little more hidden than the male, and so does not have the striking coloration. But all of this stuff is controlled by the genes that make the colors on the feathers of the bird, and each feather actually is designed to have a specific purpose when it comes to warmth and coloration and flight value, so that each part of this bird is perfectly put together. So when you look at organisms like your feline cat at home or your canine dog at home, look at those dogs and look at the coloration, look at the cats, especially the cats, because the cats are still very feral, and look at their colors and look at the way they move, and all of that's controlled by the DNA in the cells that is transcribing and translating the information to show you what we see here on the right. On the left, what you see is 2 beautiful parents and 2 beautiful children, all have inherited specific characteristics from their parents as a result of the creation of sex cells forming zygotes. How beautiful. Ho, 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 ho. Such a beautiful world of genetics. I say we move on. Why is genetics important? Well, certain people actually hold a greater risk for some cancers and tendencies than other groups as a result of their ancestry. For example, there are certain groups of Northern Europeans who have a lower risk of lactose intolerance than other peoples do, and that's because they've been eating milk products for longer as a result of needing the nutrients to survive such cold environments. Ho, 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 ho. I'm going to eat an apple right now. Chum, 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 chum. Okay, that's very good. All of this is genetics, is the pattern of inheritance. There's so much to talk about. Most of the advances in research when it comes to genetics and evolution have come from the Western European areas, where there was a renaissance of knowledge and people looking for information on how to explain the natural world. There was also a large amount of information coming from the church of the time, which had lots and lots of money and lots of time in order to think about things and why they worked and explain those things to the people. One of the most important people in the history of evolutionary theory and genetics theory was Jean-Baptiste Lamarck. Lamarck had an idea about how organisms evolved and how they changed over and over again. And what he found was that organisms can change over their lifetimes and pass these changes on to their offspring based on the need to do so. And we found that this was a wrong remembered theory, but it still helped us identify ways in which organisms over time changed. Look at the long necks of those giraffes. They look like big, big horses to me. On the other hand, we have this thing on the bottom which is crazy. It's called a megatherium. It's like an elephant-sized sloth. An elephant-sized sloth. I can't. That's amazing. Elephant-sized sloth that Charles Darwin found fossils of, and this shows that there were organisms in history, and that some of these organisms may have given rise to organisms that we see today, even though we don't have elephant-sized sloths anymore. And Charles Darwin was one of the primary people observing things in evolution. And when you think about evolution, it's these two people. Why am I talking about these two people in evolution? I'm putting the cart in front of the horse. One of the things is that both of these people were evolutionary theorists, and they were not able to explain how organisms passed on information from parent to offspring, even though it was very, very common that organisms passed on information to offspring. Neither of them knew the mechanism by which it was done. They knew how it happened, or Darwin at least did, but neither of them knew the exact discrete units that were passed on from one organism to another, or how they actually worked. My name is Gregor Mendel, and I was the first one to identify this. I was an Augustinian monk. I was born in what is now today the Czech Republic, and I was able to do lots of experiments with plants in the monastery garden in Borneo. And the best thing about this monastery was it was a center of information, learning, observation, and passing information from individuals. When I was done with this, I went through a bit of depression, and I went to take my educational teacher's test, and I failed it. So they sent me to Vienna, where I was able to work with a man named Christian Doppler, which you have probably heard the thing of the Doppler radar, which is a measurement of how certain waves, radio waves and different types of waves, are transmitted and sent back in order to get information on weather patterns and things like that. After that, I went back to the monastery to teach secondary school, and I was able to spend lots of my time working with specific types of plants, and this really showed me the interest of genetics and heredity. All right, Gregor Mendel had to go. He's a little sick, but we can talk about him a little bit. One of the crazy things about what Mendel did was he was one of the first people to kind of like manipulate the pollination and growth of plants, and he's also kind of played around with the sexual fertilization of different plant parts. What we do know about plants is that plants have two reproductive parts. They have the male reproductive parts, which are called the stamen, all right, which includes anther and filament. And what they have is the pollen grains, which are the male sperm. And then plants also have the pistil, which includes, all right, the ovaries, which houses the ovules, which are the female sex cell. So when you're eating fruit, especially like look at an apple or a blueberry or a raspberry or a pineapple, what you're really eating is the fruiting body of what was the ovaries and stamens of plants. So what Mendel did was he took the pollen from the stamens and sprinkled them on specific ovules or specific ovaries and pistils of specific plants, thereby kind of forcing fertilization of specific plants. So there are two things that he did. He took true breeding lines of pea plants, and true breeding plants are plants that only have a specific trait of a character. So a character would be like height, and some plants only had the trait tall, and some plants only had the trait short. And he kept them separate so that he had tall plants that were only giving rise to offspring that were tall, and then he would also keep plants that were short plants that were only giving rise to short plants. So he had these true breeding, or what we would call today purebreds, right? Because you have purebred dog, purebred cat, they have characteristics of only that breed and not any other breed. So he took plants that were self-fertilizers. Basically, they could fertilize themselves. They had both male and female parts, all right? And he also cross-fertilized them. He would take the genetic material from the male part of one plant and he would sprinkle it on the female part of another plant, or he'd take the female part of one plant and pollinate with the male part of another plant. So he was kind of cross-breeding and looking at what happened. So what Mendel was all about doing was tinkering with the heritability of plants using the male and the female parts of those plants. So a couple things that we need to understand before we even get into Mendelian genetics are some terms. So characters and traits are very similar, but not the same. It's important to know. A character includes things like flower color, flower position, seed color. If we're going to humans, it would be things like height, it would be things like hair color, it would be things like eye color, amount of hairiness. All right, we all know that some of us have more hair than others. And you could go on and on about what these characters can be. Traits, on the other hand, traits are forms of characters. All right. Now, if we have eye color, which is a character, the trait of that could be blue, brown, green, turquoise, whatever, hazel, different. There are different colors of the eyes. So the eye color is the character, and the trait, all right, is a form of that character. Now, traits are controlled by these things that are the actual genetic information on chromosomes called alleles. All right, so alleles are specific gene sequences that lead to specific traits of characters. So, as a character is a kind of a characteristic of an organism. A trait is a specific characteristic of an organism. An allele is the gene sequence that controls that trait, which of the character. All right. Now, some alleles are dominant and some alleles are recessive. What Mendel found was that some alleles, when the presence of other alleles, masked the other alleles. All right. He found that when you had a purple-colored true breeding plant and you crossed it with a white-colored true breeding plant, that you ended up with purple plants. And this showed actually that the purple allele, when present with only the white allele, if there was one of each, all right, then we would end up with a purple plant, and that means the purple is the dominant allele. On the other hand, the allele that is masked in the presence of the other one is called the recessive allele. And for this example, if you took a white allele and mixed it with a purple allele, if you had one purple allele and one white allele, you would end up with a purple plant. This means that the white allele was masked. All right. Also, important terms are phenotype and genotype. A phenotype is a physical characteristic, and a genotype is the specific allele combination of an organism. All right, remember that. So I may have an allele for shortness and an allele for tallness, all right? But if I'm short, it would mean that short allele is dominant. So the dominant allele doesn't have to be a dominant characteristic, meaning you would think that being tall would be a dominant character of being short, but that's not really the case here. All right, it can be, but think of genotype as the allele combination. Okay, so we're talking about homozygous and heterozygous, for example. There we go. All right, so let's say we have a character of eye color. That's our character. Now we could have specific traits of the eye color. Let's make it easy. Blue eyes and brown eyes. All right, let's say there's only two colors of the eyes, which there's not. All right, blue eyes and brown eyes. All right, now the traits of blue and brown eyes are controlled by alleles. All right, it's important to understand that one of those alleles comes from the father, and one of those alleles comes from the mother. Remember the formation of gametes, right? So let's say that blue eyes is the recessive, and brown eyes is the dominant allele. Now, when we're just arbitrarily giving one of these a dominant characteristic and one of the recessive, which means that when the big E is in the presence of the small e, we see the phenotype of the big E, which is the brown eyes. And when the small e is in the presence of the big E, the small e here, or the recessive allele, all right, which represents blue eyes, all right, actually becomes masked by the presence of the dominant allele. That's all that means. All right. So if let's say, for example, that during the process of sexual reproduction, a sperm with one big E, remember, that's why meiosis happens, because we don't want an organism donating two alleles, and if this donates two alleles, all right, then we would have four alleles. Remember, it's more common. It's most common to have two alleles of each character. Now we're going to talk about ways in which you can have more than two. But for many of our characters, many of the traits that you have and I have, they're controlled by two alleles. All right, and luckily Mendel was only working with two alleles for many of his crosses. So let's say the sperm cell has a small e and the egg cell has a big E. When we fuse, we end up with big E, small e. Now, big E, small e, this organism, if you guess what is the phenotype of this organism, all right? If the phenotype of this if you guess that this organism has brown eyes, then you'd be correct. Blue eyes is the recessive phenotype, and this blue eye allele is masked by the presence of this one. All right, and that's just dominant recessive. All right, so this organism actually is heterozygous, but displaying the dominant phenotype, which is brown eyes. All right. Now, if we change this around, let's say we have big E here or small e here and small e there, then our resulting organism would be small e, and this would be the recessive phenotype. This organism is homozygous for the recessive. All right. Homozygous just means you have the same two alleles. All right. Homozygous, heterozygous. All right.